Hello friends, how are you? This is our second video on online education. Previous video I have uploaded on Google Class. In that video I have given information regarding the how to face the online classes. Generally, up to the last or before the lockdown, we use conventional periods, conventional methods of conventional teaching methods. But due to the lockdown or COVID-19, we are turned to the online classes. Okay, there are some problems in online classes. Already I have told in first video, and how to overcome that problems also I have told in that video. Today this is the second video. It is on our syllabus. Students, first I heartily welcome one all of you in BSc second year, third semester, fishery science. Friends, in third semester fishery science have two papers, paper 6th and paper 7th. Paper 6th will teach by Airav sir, and paper 7th will be taught by me. The paper 7th title is Fish Developmental Biology. Paper 7th includes 4 units. In 4 units, 2 units related to the total developmental biology. In first unit, there are 5 sub-units or beats. In first bit, that is types of fishes based on reproduction, oviparity, viviparity and ovoviviparity. According to the reproduction of fishes, generally the fishes are divided into 3 categories, and that categories are oviparity, viviparity and ovoviviparity. What is viviparity? Generally you know that egg-laying animals are called the oviparity. Female lays eggs, either fertilized or unfertilized eggs outside the body. The unfertilized eggs are again fertilized by the sperms outside the body. In second, that is viviparity, that is generally you call live bearers animals, or that animals gives direct birth to the young ones. The embryo develops within the female body, and then the female gives direct birth to the young ones. The third category, that is ovoviviparity, is slightly deeper. In this, embryo develops inside the female body, or eggs. Embryo develops within the eggs inside the female body until hatch. After that, but eggs or that embryo lays outside the body, and eggs hatched, larva comes out. Then metamorphosis takes place of that larva. There are 2 types of eggs hatching takes place in oviparity. First, the eggs that remains in mother body until they are ready to hatch, and after that hatch, 2 type of larva comes. There is small larva without metamorphosis or complete larva. or that is metamorphosis. Meta means change. Morphosis means externally. The larva is a complete larva, or after metamorphosis the larva forms full complete, or that is called complete larva. The second bit is gametogenesis in fishes. Under that, oogenesis and spermatogenesis. In oogenesis, how the ova formation takes place within the ovary, and spermatogenesis, how the sperm formation takes place within the testis. We have to learn about that oogenesis and spermatogenesis. Number third unit, under this third unit, we are going to learn about the how many types of eggs. Types of eggs, there are different types of eggs according to the size, according to the yolk membranes, according to the position of yolks. So there are different types of eggs are present in fish, and what type of eggs, what type of their classification we will see under the types of. Now fourth unit is the fertilization of eggs. You know, already I have told that there is oviparity species lies eggs, either fertilized or unfertilized eggs. The unfertilized eggs, when lies outside the body, the sperm's union of gamete to gamete takes place. That is eggs and sperms, and eggs get fertilized. And after that fertilization of eggs, the fifth subunit is, that is cleavage starts. The cleavage is nothing but there is segmentation of cells within the eggs. After the fertilization eggs, there is nothing but there is an embryo, small embryo. In that egg, the embryo develops. That is, cleavage of cells or division of cells starts, and after that, the second unit is start, and that is called the developmental biology. In developmental biology, there is again 4 units are there, or 5 units are there. See here, the first is a morula, second is a blastula, third is a pet map of blastula, fourth is a gastrulation, and fifth is a hatching and post-embryonic developments. After cleavage, there is a segmentation of cells within the eggs takes place. The process of segmentation continues, and early stage the morula is forms. The morula develops continuously. The next stage is called the blastula. The changes takes place within the embryo, developing embryo in the egg, and this stage is morula blastula occurs, and after that fate map of blastula is forms. Fate fate with future. What type of animals or formation takes place within the embryo is given the idea, that is called the fate map blastula, and after that gastrulations is. In the gastrulations, again the animal develops within the embryo, the particular stage occurs, and after that the body forms takes place, or different organs are forms within the embryo, and larva stage forms within the eggs. After that, hatching takes place. If the larva complete forms within the eggs or embryo, the egg hatched out there. The position of hatching of eggs or breaking of eggs, that is called the hatching. After formation, complete organism within the eggs or larva, they try to come outside the eggs by breaking the covering of eggs, hard covering. That is called the hatching. And after that hatching, when the larva comes out, there is changes takes place in their body, in the animal, that is called the post-embryonic developments. Generally, the fishes passes through the different stages. There are the semi-fry, fry, semi-fingerlings, fingerlings, seven eyes, and adults. These are the post-embryonic developments occurs in the fish. The third unit is a reproductive biology. Under the reproductive biology, there are again 5 sub units. First is the sexual dimorphism in fishes. How can we? Generally, you know that fishes do not have a sexual dimorphism characters. Generally, you cannot distinguish externally either male or female. But in few fishes, we can easily identify with particular characters which are present outside the body, and that is called the sexual dimorphisms characters. With the help of these characters, how can we identify either it is male or female? The second subunit is the parental care in fishes. Generally, you know that fishes feeding takes place at one place and breeding takes place at another places. After the breeding, immediately they lose their spawning ground, and again there comes to the feeding grounds. They never take care about their eggs or offspring. Only the spawning takes place at spawning grounds. But in few fishes, the parental care is develops. How many fishes, what type of the parental care takes, either by male or female, we are going to study under the parental care. Number third, maturity stages in male and female. After juvenile stage, when the fish attain first maturity. So for the attaining the maturity, what changes takes place inside the gonads? How many stages are occurs in the gonads? And by the stage of the gonads, the male and female identified, or it is called it is a mature fish or immature fish. So generally, fish passes through the different stages. The gonad passes through the different stages. That is immature, maturing, mature, ripe, and spent. These are the stages of maturity of fish. After that, attaining the maturity, spawning it takes place, and that spawning, the the gametes expel outside the body. In female, the egg laying outside the body, and that is called the fecundity. How many eggs are laid by the female in total spawning season or one spawning period? That is called the fecundity, and that fecundity can be estimated by 3 methods. One is the volumetric methods, the gravimetric methods, and third one is the von Bayer's method. By 3 methods, you can identify, you can assessment, you can estimate the fecundity of fish. Fifth subunit is the study of gonadosomatic index. Study of gonadosomatic index, gonadosomatic index, it is related with particularly, that is a fish body weight and weight of gonad. The relations in between body weight and weight of gonad, it gives the GSI, that is gonadosomatic index. It is very important in identifying in particular spawning phase or spawning stage of fish. When the gonad increase, the weight of fish increases. There is a correlation in between size of gonads and weight of fish. The fourth unit is the growth studies. Generally, growth of fish after taking a birth, animal, when the time is passed, the animal grows, and that growth, generally you know, the food is converted into the bodybuilding. Food is converted into the bodybuilding, along with the age of fish, growth of animals takes place. What are the growth, how it is attending the growth, the period of growth, all this we will study in the growth. Second, factors affecting the growth in fishes. Growth of animals is totally depends upon the environmental factors. Same species of same genus having a different growth rates in a different locality. It is because due to the availability of the environmental factors. The environmental factors is directly and indirectly affect on the growth of fishes. What are the important factors? How they are affect on growth? All they are we are going to study under the factors affecting the. Third is the ponderal index. The ponderal index also called the conditional factors, and it is used for obtaining the information on seasonal variation of the conditions. Seasonal variations of the condition of fish in relation to the variations. What are the seasonal variations occurs? Particularly on animals, we are going to study under the ponderal index. Length width relationships. There is a particular relation in between length and weight of fish. Generally, animal grows on particular at the particular age, and after that, the growth of animals is stunted. The weight and length is having relations, and what their relations again we will see in the length weight relationship. Number fifth subunit of fourth unit, that is the methods for age and growth determination of fish. You know, the fish which lives in water, and it is very difficult to the assessment of the growth of the fish. Those fishes are wandered or present in sea waters or river waters. The pond fishes can be, the growth of fish or age of the fish can be determined easily in stagnant water or in pond water, but it is very difficult to determine in running water or sea waters. But in fishes, the growth and age can be determined by 4 to 5 methods. One is the direct methods, which is generally used in stagnant water. Second method, tagging methods. Tags are used, particularly it may be plastic, or it may be fiber, or it may be metallic. They are used tag for determining the age and growth of fish. Next method is marking method is also available for determination age growth. Counting rings on hard body parts. Already you have studied in BSc first year, particularly the scale methods. The scale, on that scale the particular marking is available, centrally focused, circularly, annually, and that is are very useful for the counting the passing age of the fish or past age of the fish. Next is the radio carbon uptake method, is also very good methods for determining the age and growth fishes. And last is the RNA DNA ratio methods. So all these are methods for age and growth studies. We will see in at the time of age and growth studies. This is about the syllabus of BSc second year, third semester, paper number 7th. Next video I will start the types based of fishes on reproduction, and I will clear all these points about this, and also we will provide the notes, and that notes I will either send on WhatsApp group, or I will post on the Google Class. Okay, see you again.